Okay, so you are getting my voice? Hello?
Yes, sir.
sir.
Yes, sir.
Okay, okay, okay. So in last class, I have discussed about the metabolism of carbohydrates. So some part of that class is left. So today I will continue from that only. So you know the glycolytic pathway, how the process and how metabolism takes place in the body. And there are different pathways. By that way, like glycolytic pathway, TCA cycle, pentose phosphate pathway, and how the glucose is converted as a, or how glucose is used as a source of energy, galactose is used as a source of energy, glycogen as a source of energy, propionic acid as a source of energy, and acetic acid, like measure the acetic acid, measure volatile fatty acids. So how acetic acid used as a source of energy? So this is all about the energy sources, how they are metabolized in the body. And so you are well aware about that, the glucose as a source of energy. When we talk about that, so the process of glycolysis, you are knowing, all of you are knowing the process of glycolysis and where it can take place. So it takes place in the cytoplasm. And during the process of glycolysis, it forms two pyruvate units and pyruvate which can be enter into the mitochondria. And thereafter, after entering into the mitochondria, the acetyl coenzyme is produced in citric acid cycle or TCA cycle. And further, there are generation of high energy bond during the catabolism of glucose. So here you can see that in this table, the ATP yield from the complete oxidation of 1 mole of glucose. This is also you read in biochemistry as well. This is similar things. So in glycolysis, the glyceraldehyde 3-phosphate dehydrogenase, where 6 ATP, and phosphoglycerate kinase, that is the oxidation at substrate level. So we are talking about the high energy bond production. So oxidation at substrate level, 2 ATP, pyruvate kinase. The oxidation at substrate level is 2 ATP. So total 10 ATP. But ATP consumption by the hexokinase and phosphofructokinase, that is the minus 2 ATP takes place. Or you can say that the 2 lactate in the anaerobic pathway. So net ATP production is 8. Like in case of citric acid cycle, net ATP production is 38. And total per mole of glucose under aerobic condition, you have to mention that under aerobic condition, that will produce 38 ATP, and total per mole of glucose under anaerobic condition is 2 ATP. So here you can see that the NADH, which is formed in glycolysis, is transported to mitochondria. And the cycle of transportation is known as Maillard pathway. And if glycerophosphate pathway is used, then only 2 ATP would be formed per mole of NADH. And the total net production of ATP is 36 ATP instead of 38 ATP. So this is a special circumstances. During that condition, only 36 ATP are being produced total per mole of glucose oxidized. So you go through this table, and this table is available in most all the books. So it will be better to read those books as well for clear understanding. So I will not discuss much about this, like fructose as a source of energy and galactose as a source of energy, but I have to discuss about the glycogen as a source of energy, how it can be utilized. Okay, so glycogen, which is a major form of storage carbohydrate or animal starch, we can say that it is also known as animal starch. And the breakdown of the glycogen which takes place within the cell. And further, it catalyzes the hydrolysis of 1,4-glycosidic linkage. You can see in this figure. And thereafter, the glycogen and release of the glucose 1-phosphate, as well as certain amount of extra glucose is also being produced. So glucose 6-phosphate, which will enter for glycolysis via pentose phosphate pathway. So the production of glucose 6-phosphate from glycogen, which does not involve expenditure of ATP. This is very much important. The production of glucose 6-phosphate from glycogen doesn't involve expenditure of ATP, except some conversion of residual glucose to glucose 6-phosphate. So the energy production from glycogen is slightly more efficient because the expenditure of ATP is not involved there. And so energy production from glycogen is slightly more efficient than from glucose. Now, next is how propionate and acetate converted to glucose. So propionate, you know, it is as a source of energy, and in case of ruminant, it is well established that propionate supplemented most of the source of the energy. And conversion of succinyl-CoA is the major things, and so conversion of succinyl-CoA then enters into TCA cycle, which is then converted to malate. You can see here in this figure, and malate, which is transported into cytosol again, and it is then converted into oxaloacetate and further phosphoenol pyruvate PEP to fructose diphosphate. So like that, the propionic acid can be converted, can be used as a source of energy, and finally they converted to glucose. And per mole of propionic acid, how much energy is produced? So 13.5 mole of ATP is being produced per mole of propionic acid. You must remember this value, or you can note down the per mole of propionic acid produces 13.5 mole of ATP. How acetic acid can be used as a source of energy? So acetate or acetic acid is one of the major volatile fatty acid, and 65% of the volatile fatty acid which contains acetate. Ratio I have mentioned in last class. What is the ratio of acetate, propionate, and butyrate? So acetic acid is one of the major volatile fatty acid which is being produced during carbohydrate digestion in ruminant, especially in case of ruminant. And acetate, which is only volatile fatty acid, which is present in significant amount in peripheral circulation. So you must remember that this question always coming in the examination, which volatile fatty acid is present in significant amount in peripheral circulation. The answer is acetate. So acetate is converted to acetyl coenzyme A by the action of acetyl coenzyme A synthetase enzymes, and you can see in the detail in this figure. And the formation of acetyl CoA which takes place in the cell cytoplasm, and further oxidation via TCA cycle, which is limited to the mitochondrial matrix. So TCA cycle, which is oxidation via TCA cycle, is limited to the mitochondrial matrix, and further acetyl coenzyme A is unable to cross the mitochondrial wall, and which is to be complexed with carnitine, when you go through when you study the book or read the books, then you can see the details how this acetate is converted to. But you can see in this figure also. So complexed with the carnitine, and within the mitochondrial matrix, this complex is further broken down, and that releases acetyl CoA, which then enters into TCA cycle. So TCA cycle, when enters, they oxidize to yield 10 mole of ATP per mole of acetate. So you should remember this data. So per mole of acetate producing 10 mole of ATP. And two high energy phosphate bond which are being used in the initial synthetase mediated reaction. So net yield of ATP 2 mole is further utilized. So net yield of ATP is 8 mole per mole of acetate. Like butyrate, formation of acetyl CoA from butyrate, how it happens. So butyric acid, how butyrate is as butyric acid is being used as a source of energy. So initially butyric acid, which is converted to beta hydroxybutyrate, and during absorption across the ruminal wall, or not only ruminal wall, also a omasal wall. So during absorption across the ruminal as well as omasal wall, they are used as a source of energy by different body tissues. So beta hydroxybutyrate, which is converted to acetoacetate, and then acetyl coenzyme A via succinyl coenzyme A pathway, and finally acetyl coenzyme A is formed. And detail you can see in this diagram, which is also available in the books. You can read the books to get more clarity. And further, acetyl coenzyme A is metabolized via TCA cycle, and ultimately glucose is being produced. And you should remember the data. Net gain per mole of butyric acid is 22 ATP. Net gain per mole of butyric acid is 22 ATP. So highest energy between these three volatile fatty acid per mole gain from the butyrate. So this is about the carbohydrate metabolism, and you can read detail in book, and if you have any confusion, then you will ask in coming class about this today class. So in coming class, I will discuss about the fat metabolism, either fat or protein metabolism, any one of them. And you will get the slides presentation and I will share in the group this PDF of the slides.